Welcome back to the course Fundamentals of General Immunology. So we will start today our module 6. Title of the module 6 is Immune Effector Programming, and we'll discuss that in that module about transcription control of the immune effector functions. That means we'll try to understand what are the transcription regulation that's happening in case of a cell which undergoes this kind of activation either in the T cell or in the B cell. So as we know that many of the gene regulation in a cell is controlled by various factors, and these factors can be environmental or any stimuli which can lead to various type of changes in the cellular environment that could lead to the change in the transcription pattern of a gene. And if there is a transcription pattern change, or if there are different kind of mRNA which are produced post transcription, eventually what we will see is a change in the phenotype of a cell. Or particularly in case of immune cell, we see a lot of regulation of the protein that is happening, which can lead to different function of an immune cell. So let's try to understand about this transcription control of immune effector functions. So the core concept about this transcription control is these are actually that immune cell functions are regulated by transcription factor that turn gene on or off. That means when there is a requirement for any particular protein, we see that the genes are getting transcribed to produce that protein. And if at all this is not required, then probably we see that those gene transcriptions are getting off. We will categorically see what are the genes which are getting turned on and off in case of B and T cell population, particularly in context of the immunoregulation. Again, it defines the effector molecule secretion, particularly in terms of cytokine, enzymes, receptor expression, different kind of CD molecules which we have discussed. For example, we know that there are cytokines which are produced such as IL-2. There are some kind of receptor which are produced on the cell surface such as CD28 which are expressed in T cell population, and that's very important and critical for co-stimulation. How this regulation of protein is happen in case of the gene level, like how these are transcriptionally controlled, is mostly depend upon the stimuli of a cell type, which cell type is receiving what kind of a stimuli can decide what kind of gene are getting turned on or off. So let's see it in detail, that what are the things which are happening particularly in context of B cell. The first we will try to cover B cell, and then we'll go for the T cell. Now keep in mind that the transcription regulation of B cell and T cell are entirely different, because B cell as such, the phenotypic feature of the B cell is entirely different as compared to the T cell. B cell is actually going to produce a lot of immunoglobulins. They can produce some kind of a memory immune response which eventually lead to production of a future different immunoglobulin chains which are responsible for other immune regulation. So we'll see both these cell types differently. So let's try to concentrate first on the transcription control of B cell function. So if you focus here that we have a BCR, which is, again I'm re-emphasizing multiple times, when it comes to BCR, which is called B cell receptor, actually it's an immunoglobulin Ig molecule, and this Ig molecule is actually going out and doing some function from time to time. Apart from that, these B cells are having other receptor as well. In case of the T cell, we have the different kind of ligand like 40L ligand, cytokine, different kind of cytokine which we have discussed. Now any of these molecule, when there is an interaction of an antigen antibody, this could lead to the proliferation and controlled transcription expression in the B cell. Let's try to understand the different kind of signal transduction that happening in the B cell, and we'll go one by one. So the first one is NF kappa B pathway. Now this is nuclear factor of kappa B pathway of the B cell. So this is called nuclear factor of kappa B cell pathway, which is actually a very specifically transcription factor which can be activated on B cell. Then we have PI3 kinase and AKT pathway. It's a combined pathway, again a very important pathway in cancer biology. We'll see how these can modulate a B cell expression or different gene expression in B cell. Then we have mitogen activated protein kinase pathway, which is also called as MAP kinase pathway. And then finally, JAK-STAT pathway. Now, Janus kinase and signal transducer and activator of transcription pathway is very important pathway for regulating interferon production in innate immune cells. But here in case of B cell, we'll see how these are responsible for producing different cytokines, because regulation of the cell it depends upon different kind of cytokines as well. So in general, we'll try to focus on NF-kappa B, PI3 kinase pathway, MAP kinase pathway, and JAK-STAT pathway one by one. So these are signaling molecule which can activate some other molecule. Let's see what are those molecules which are getting activated. So these are doing some activation of the transcription factors such as PAX5. PAX5 stands for paired box 5 protein, which is maintaining the B cell identity and survival. So these can help the B cell to survive little longer. So we need to express PAX5 protein if we want to make the B cell little longer into the circulation. We know in general the lymphocytic cell life is 7 days in blood. In some cases it may be lesser, some cases it will be higher. It's not like it's completely 7 days, but we can see a variation of plus minus few days. But in general, if we want to make a little longer survival of the B cell, we need to activate some of the genes which are responsible for making survivability a little longer. And one such gene is PAX5. The second is BCL6, which is actually a B cell lymphoma 6 protein. BCL6 is actually help the B cell proliferation. It promotes the germinal center formation and also help in more and more proliferation of B cells. So this is also a very important protein which is required for the B cell activation and proliferation. Third one is BLIMP1, which is B lymphocyte induced maturation protein. If you critically look into the name, it's called B lymphocyte, which is actually a B cell induced maturation protein. So basically it helps in the maturation of B cell, and it can be derived plasma cell differentiation. One such factor which is noted for this protein is it can downregulate the PAX5 expression. That means if we have a more expression of BLIMP1, we see that there could be less production of PAX5. So there could be a protein inhibition by the expression of other proteins. So this is one such protein which is responsible for cell differentiation. Fourth one is IRF4, which is interferon regulatory factor 4. Again, IRF are multiple different types of IRF, such as IRF3, IRF7. These are involved in apoptotic processes in the cell. IRF is also responsible for different interferon secretions in the cell. But particularly in context of B cell population, it activates plasma cell gene program and survival. So basically it also helps you to survive the cellular mass of B cell in the circulation. Now if we talk about the survivability, we need to understand that definitely the process will lead to anti-apoptotic activity. So the proteins which are responsible for programmed cell death will be inhibited because of that. Because ultimate function is we want to survive that cell a little longer in the circulation, and therefore we need to inhibit the apoptosis. So some of the anti-apoptotic gene will be activated. The last one is XBB1, which is X-box binding protein 1. Again, it induces the ER expansion. ER stands for endoplasmic reticular expansion. And once we see that the ER expansion is happening and all, we can see all the regulation which are related to ER, or say post-translation modifications, stuff like that, will be controlled by XBP1 expression. And of course, it can maintain the antibody secretion in a proper way. So these are some of the key factors which are responsible for transcription regulation in the B cell: PAX5, BCL6, BLIMP1, IRF4, and XBP1. Again, we need to just remember that these are the transcription factors which are very critical for B cell activation pathway. This will change once we'll discuss about the T cell population. So all right, we'll move on and we'll see that this kind of gene expression can do multiple different outcome in a B cell population. First will be a proliferation. Of course, this will lead to the expansion and proliferation of the B cell population. It can also stimulate the germinal center, which we have discussed in the BCL6, that it can do a high affinity B cell production in the germinal center. We can also understand that this can do a class switching phenomena, which we have discussed, that there could be a class switching phenomena from IgM to IgG. Yesterday we discussed about that the proliferation of a particular kind of cell eventually in the long term can do a class switching phenomena where the heavy chain of one particular immunoglobulin will be changed to the other one. And we notice that this is very general statement when we talk about class switching, that IgM can change to IgG. That means a mu chain can change into gamma chain. And therefore, this class switching phenomena is also very important when it comes to specifically B cell regulation in terms of transcription control. Because these are the sites, these are the factory where B cells are produced or manufactured. Again, the plasma cell differentiation in terms of memory immune response, we need to understand that plasma cell is also critically controlled by transcription of multiple genes which allow the cell to survive longer duration, because eventually the memory cell you want to stay a little longer in the human body. Say there are antigens for which we get a very long lasting immune response. For example, pox virus is specific memory. So once a person vaccinated with pox virus will get immunized throughout the life, because the memory immune response is very robust, and therefore even if there is a relapse or any kind of encounter of the pox virus in future, the immune response can fight against those entering pathogen very effectively. So very strong memory immune response against pox virus. While in case of COVID-19, we know the memory response is very transient. So we can see a very short-lived memory immune cells, and which is not good in terms of human being, because you don't get an effective protection for the long term. So therefore, the plasma cell production and the memory immune response is very important when it comes to long-lasting immunity. And also, again, the same thing, the long-lasting immunity when we have a memory cell, which are important for survival and secretion of the antibody in the longer duration of the time. So this is all about transcription control of the B cell. These are the four pathways which we'll try to see individually. We'll see first NF-kappa B pathway, and then we'll go for PI3 kinase pathway, MAP kinase, and JAK-STAT pathway at the last. So let's move on to NF-kappa B pathway. So this is a very important pathway in terms of activating a B cell. So we know that nuclear factor of kappa beta, as name itself indicates, is very specific for B cell population. So what is happening in the first case, that we could have an antigen bound to an immunoglobulin chain or the other B cell receptor which are present on the membrane of a B cell. We could have a CD40 and 40 ligand signaling pathway, and finally there could be a way where toll-like receptor, which are the surface protein that can bind to pathogen associated molecular pattern, which is called PAMP, which are coming from outside. So the toll-like receptor or CD40 signaling or the B cell receptor pathway can lead to different kind of downstream signaling. So we know that MID88, this is an adapted protein in the toll-like receptor, can be activated upon binding to pathogen associated molecular pattern. This is again very critical. Many times this can be asked in the examination, that what are the downstream adapted molecule which can be activated post binding of a ligand to toll-like receptor, and the answer is MID88. And then CD40 and 40 ligand can do an activation of TRAF2, 3 and 6. There are multiple TRAF protein which are getting activated, and TRAF stands for here TNF receptor associated factor, which are very again critical, because that can be regulating the tumor necrosis factor specific protein. And again we know that the cytoplasmic domain of these proteins which are there in B cell receptor called ITAM, which will be phosphorylated by different kind of kinases present in the cytoplasm. So anyway, after binding to any of these antigen specific complexes to the B cell receptor by either CD40 signaling or toll-like receptor or any other protein, it can lead to some activation cascade which can lead to adapted protein. Now what will we eventually doing it? So there could be an activation of two distinct kind of pathways. One is called canonical pathway and the other is called non-canonical pathway. Remember, the canonical and non-canonical pathway will be entirely different in terms of their phosphorylating protein. So we need to differentiate both canonical and non-canonical pathway in terms of which proteins are getting phosphorylated. So let's try to focus on canonical pathway first. So this is a very rapid activating pathway, very fast pathway. So what is happening that here we can see that inhibitor of kappa beta alpha. So we have discussed that the NF kappa B in the cytoplasm can bind to its inhibitor. So actually the NF kappa B is actually a complex. It's an heterodimer of P65 and P50. So for time being we can understand that these two is actually NF kappa B, and this NF kappa B is heterodimeric form of P65 and P50. These are in the bound form with its inhibitor, which is called inhibitor of kappa B. Now this inhibitor can sequester the NF kappa B in the cytoplasm. Once this inhibitor is getting phosphorylated, then we can see that this inhibitor, which is actually sequestering the NF kappa beta in the cytoplasm, once it is getting phosphorylated, this can go for ubiquitination. So this inhibitor, which is bounding this NF kappa beta in the cytoplasm, once it is getting phosphorylated by some kind of an kinase complex which are there in the cytoplasm, then this inhibitor can go for degradation. So this will undergo proteasomal degradation. Once this is degraded, then this NF kappa B, which is a heterodimer of P30, P65 and P50, become free to go to nucleus. And since the ultimate goal of this transcription factor is to go to the nucleus to start transcription of certain genes, will happen once they will go to the nucleus. And to go to the nucleus, they need to be free first. So here we need to remember, in the canonical pathway, this inhibitor of kappa B alpha is getting phosphorylated. Even we can just say that inhibitor of kappa B can be getting phosphorylated, and after phosphorylation this will go for ubiquitination. Ubiquitination is a process where this very specific cellular organelle called proteasome is degrading all the proteins into smaller fragments. So this I kappa B can go inside this tunnel-like structure, which is called proteasome, and will degrade into smaller protein fragment, and the process is called ubiquitination. This can be done by binding to different kind of universal protein which are present in cytoplasm called ubiquitins, and this process is called ubiquitination. So we will not go in detail about that. So what is evident that after phosphorylation, this protein is getting degraded. Now once it is degraded, then this NF kappa beta, which is a heterodimer of P65 and 50, become free to go to the nucleus. So this is canonical pathway. This is a very rapid pathway. Let's move on to non-canonical pathway, which are happening very delayed way. It's a kind of a very slow process, and that's happening in case of non-canonical pathway. And what's happening here, that we have this different protein coming into picture. So this is called P100. So this P100 is getting phosphorylated by again different kinases, and once this kinase is phosphorylating, then this P100 is getting degraded partially to release P52. It's a part of P100 molecule. When it comes to P100, P52, 65, 52, like that, these stands for the KD of the protein. P65 is mostly the 65 kilodalton protein in general. P50 stands for 50 kilodalton protein. Here also the P100 is 100 kilodalton protein, which is getting dissociated post phosphorylation into a smaller one, P52. Once P52 is getting dissociated, then there could be another protein called REL-B, which is coming and bounding and getting a specific, say, combination with p52 to become NF-kappa B. So this is actually nuclear factor of kappa B that will go to the nucleus and activate the pathway. So in canonical pathway, we get phosphorylation of inhibitor of kappa B. In case of non-canonical pathway, we get phosphorylation of p100. In case of canonical pathway, the release factor is p65 and p50 heterodimer. In case of non-canonical pathway, where we have rel B and p52 heterodimer to go into the nucleus. So this is two distinct pathway in NF-kappa B. Now we can see what are the things which are happening. So these are the full forms which are there, such as B cell receptor, co-stimulatory receptors such as CD40, toll-like receptor. These are inhibitor kappa beta kinase complex, and this could be alpha beta and NEMO, which is also called as IKK gamma. Then we have inhibitor of kappa beta alpha, NF kappa beta, of course. These are canonical and non-canonical, which are listed here. So let's try to see what's happening in case of both canonical and non-canonical pathway. Of course, that will go and bind to the genes which are located in the chromosome, particularly which can start doing a gene. So basically this can go and bind to kappa B site in target gene promoter and enhancer, and therefore it can cause transcription of different genes. And these transcription of genes can lead to various function of B cell. One is proliferation, and again we will repeat this sentence multiple times in this whole pathway because this is what the function of B cell activation. So we need to understand that how it can cause B cell proliferation, how it will allow B cell to stay little longer in the plasma cell, how the memory cell response will be little enhanced because of these all canonical pathway and non-canonical pathway. And of course, when I said that it will allow the cell to survive a little longer, and when we say that it will allow the cell to survive longer, we need to understand that there could be an upregulation of anti-apoptotic gene which will not allow the cell to die. And some of the genes are BCL2, B cell lymphoma 2, B cell lymphoma XL, and MCL1. So these are very specific anti-apoptotic genes which are getting upregulated upon NF-kappa B activation or pathway. Apart from that, we'll get a lot of cytokines which are produced, such as IL-6, IL-10, TNF-alpha, BAF, etc. So these are all different kinds of cytokines which are helping B cell proliferation, survival, and secretion of different cytokines. Again, differential antibody response, we can see that where we could see different kind of class switching phenomena and germinal center production of different plasma cell differentiation, and also the very effective memory immune response prolongation. So essentially this whole pathway we need to critically look into how these are activated, and the pattern which we have to remember is what are the genes which are getting phosphorylated and how this can lead to transfer of the signal to the nucleus. Ultimately we could see that there is an heterodimer in the canonical pathway, non-canonical pathway, and just remember that these two are different in canonical and non-canonical pathway because of the heterodimeric protein associated with it. So this is all about NF-kappa B activation pathway in B cell, and let's try to move on the second one, and the second one was the PI3 kinase AKT pathway. So this is again a very important pathway. All the four pathways are important, some are critical in a particular time, and these all depend upon what kind of a stimuli they are getting. So of course the same thing is here. We could see the activation cascade is going on when there is a binding of antigen to B cell receptor, also the co-stimulation by CD40 and 40 ligand, and this could lead to multiple different kind of events. And one such thing which we could look into is how this PI3 kinase, which is called phosphatidyl inositol 3 kinase, this kinase is getting activated, which can convert this membrane bound PIP2 into PIP3, phosphatidyl inositol diphosphate to triphosphate. So once there is a phosphorylation, that means there is an involvement of ATP, which can cause addition of phosphate group to the PIP2 to make it PIP3, and then adenosine diphosphate will be released into the media. which can activate the AKT. Now, a little bit detail about the AKT is given here. The AKT kinase stands for RAC alpha serine threonine protein kinase, and it is commonly referred to protein kinase B. In general term, we generally say protein kinase B, and the name of AKT derived from a very specific virus which is called AKT8 retrovirus. So retroviruses are the viruses which can cause HIV-like symptoms. So one of the retrovirus is what is human immunodeficiency virus, and among all these retroviruses, there are retroviruses which can carry different oncogenes, and one such oncogene is called viral specific AKT, and from there this name is derived there. AKT activation. This is a very specific kinase which can cause different kind of transcription factor activation, and NF-kappa B is also one of them. Apart from that, there are other transcription factors such as FOXO1, CREB, which stands for again different kind of cyclic AMP response factor, then BLIMP1 and XBP1. So these are kind of transcription factor which are, say, mixture of one way or other way, but ultimately this can help to survive the B cell population, proliferate, differentiate, antibody production, and cytokine release. So PI3 kinase, AKT kinase pathway can also be doing the similar kind of function by activating similar kind of protein such as BCL2 and BCLXL, which is anti-apoptotic gene. So just keep in mind these two proteins, once there is an upregulation, we can see the cells are surviving little longer. That means when the cells need to survive little longer, we need to express this protein into cell type. So these are anti-apoptotic proteins, BCL2 and BCLXL. There are cycle-independent kinases, c-MYC, and these are helping in proliferation, because once we have a B cell, we want more and more B cell to be there before class switching phenomena and stuff like that. So in these cases, we have c-MYC or cycle-independent kinases to be activated. Apart from that, we also need differentiation of the cell type, more and more antibody production. Then we can see a lot of IgG gene recombination and other processes which are happening in case of B cell. These are all done upon the activation of PI3 kinase pathway and AKT pathway. So this is the combined PI3 AKT pathway. So then we will move to the third one, and the third one is MAP kinase pathway. Again, in a very similar way, we can see that once there is an antigen binding to the B cell receptor or for the co-stimulation, we can see that there could be an activation of B cell. So now we have understood that the B cell can be only activated when there is a co-stimulation with CD40 ligand or by binding to an antigen. And once there is a binding, we can see that the MAP kinase, which is called mitogen activated protein kinase pathway, can do a phosphorylation event of a series of molecules, RAS, RAF, MEK, and finally ERK, which is actually leading to a MAP signaling cascade. So we can have an activation of transcription factors such as activator protein 1, ETS-like 1 protein, and finally the CERB protein, which is cyclic AMP response element binding protein, which we have seen earlier also. So here just a correction here, so this is not CERB, but this is CREB. So this is cyclic AMP response element binding protein that can help in transcription of multiple genes which can lead to the cell survival, proliferation, differentiation, antibody production, and cytokine production, similarly like PI3 AKT pathway. That means what is changed here is the kinase. So once we have a PI3 kinase can do a downstream activation of different transcription factor, we see the effect. If there is a PI3 kinase, AKT kinase activation, we can see a different pathway altogether. But the whole purpose of this transcription factor activation is these 5 fundamental processes which are happening in B cell: survival, proliferation, differentiation, antibody, and cytokine productions. So we have to look this in a very critical way, that ultimately it can lead to these 5 basic processes in the B cell. And finally, the last pathway is JAK-STAT pathway. We'll try to see this JAK-STAT pathway later on again when we try to see interferon production. Now, JAK-STAT pathway is a little different in terms of its activation, because this can activate upon the binding to the cytokine. So once there is a cytokine binding to the cytokine receptor present on the cell membrane, B cell, then we can see that there are proteins such as Janus kinase. JAK stands for Janus kinase. There are many isoforms of Janus kinase in the cell, such as JAK1 and 2, and then the ultimate thing which is happening in the downstream cascade is the phosphorylation. So JAK becomes phosphorylated. Phosphorylated JAK will phosphorylate STAT protein. Now, STATs are also very different kind of STATs are present in B cells, such as STAT6 and STAT3. So there are many other STATs which are present, and generally STATs are before going to nucleus, they become dimerized. So once we see that the STAT become phosphorylated, we see that the phosphorylated STAT become dimerized. And once these are become dimer, then they can migrate to nucleus, and then they can start transcription of the gene, which can lead to activation, exactly the same 5 phenomena which we have discussed in the other pathways as well. So the difference between the JAK-STAT pathway and the other pathway is the way of activation. Here we can see that different kind of cytokines such as IL-4, 6, 10 and 21, which can activate the cytokine, the production of JAK-STAT pathway by binding to the cytokine receptor. So once they can bind to cytokine receptor, there are proteins which are present in the cytoplasmic tail of the cytokine receptor called JAK, and there could be multiple isoform of JAK, and once the JAK protein is getting phosphorylated, this can phosphorylate the STAT protein. So therefore this pathway is called JAK-STAT, because JAK is always doing phosphorylation of the STAT protein. Apart from many other phosphorylation events, this is very well explored pathway. And once the STAT is getting phosphorylated, there could be a dimerization. So a similar kind of a STAT, when they bind, called homodimerization, sometimes it becomes heterodimerization as well. So once this STAT is getting phosphorylated, a dimer form of the STAT can go to the nucleus. So there is a very obvious form of question which can be asked, that non-phosphorylated form of the STAT is located in cytoplasm, and the phosphorylated form of the STAT is located inside the nucleus. So we could understand that phosphorylation is a critical event which allow the protein to go inside the nucleus. Before that, you can see that all the proteins are in the cytoplasm. So once phosphorylation happens, then the protein can migrate inside the nucleus. So if we do fractionation in the molecular biology, which generally we used to do fractionation, say if somebody asks you that how you can segregate the phosphorylated form of the STAT to the non-phosphorylated form of the STAT, we can simply do the fractionation of the cytoplasmic fraction and the nuclear fraction separate, and we could identify all the phosphorylated form of the STAT in the nucleus, while the non-phosphorylated form will always be there in the cytoplasm, because these are the critical compartment which allow differentiation of these two cells, and immediately after phosphorylation this can go inside the nucleus. And phosphorylated STAT is a very strong transcription factor that can bind to the genes and start transcription, and therefore these are called signal transducer and activator of transcription. And once we see the activation, we can see that multiple different kind of cytokines and genes are getting proliferated, which can lead to the critical 5 processes which we have discussed: survival, proliferation, differentiation, and production of antibody and cytokine. So this is all about, in nutshell, about the B cell activation cascade, where we looked into 4 critical pathway: NF-kappa B, PI3-AKT pathway, MAP kinase pathway, and JAK-STAT pathway. And these are all 4 very critically controlling the expression of the gene, which can lead to survival of the B cell into the atmosphere to give a proper immune response to the human being. So that's all about the B cell transcription activation. We'll try to see about the T cell transcription control in our next class. Thank you.